Dear conference participants, let me present a report to you, Accumulation of endocrine-disrupting compounds in Procambarus Virginalis in Dnipro River, ecological and hygienic aspects. The author of project, Oleh Lykholat, University of Custom and Finance, Dnipro, Ukraine, Oleh Marenkov, Olexandr Dnipro National University, Ukraine, Oleh Nesterenko, Olexandr Dnipro National University, Ukraine, Tetyana Lykholat, Olexandr Dnipro National University, Ukraine, Maxim Kvitko, Yuriy State Pedagogical University, Ukraine, Olexandr Kobryushko, Yuriy State Pedagogical University, Ukraine, Yuriy Lykholat, Olexandr Dnipro National University, Ukraine. Next slide, please. Ecosystem pollution is a serious problem worldwide. Aquatic ecosystems are extremely important for biota, fauna, flora, and humans. In recent decades, there has been a worldwide concern for the environment due to the difficulty of removing pollutions through traditional water and wastewater treatment processes. Among environmental toxicants, endocrine-disrupting compounds encompass nearly 800 different chemicals, including both natural and synthetic compounds, including pesticide chemicals in consumer products and food contact material, pharmaceutic and personal hygiene products. Next slide, please. The marbled crayfish Procambarus Virginalis belongs to freshwater crayfish species. Procambarus Virginalis is a purple pet species in Europe and North America. Procambarus virginalis is an invasive species in the Dnipro River in Ukraine. Now the species has been introduced into the natural ecosystems of the Dnipro near to the city of Dnipro. Through the parthenogenetic methods of reproduction and polytrophic omnivorousness, Procambarus virginalis can withstand low water winter temperature in the European temperate zone. Next slide, please. Studies indicate the need for monitoring to assess certain classes of pollutions, such as EDCs, which are known to release serious toxicological hazards to aquatic biota at low concentrations. The purpose of this work was to determine the concentration of hormones, mainly estradiol and cortisol, in Procambarus virginalis of different age as hydrobion from the Dnipro River in the city of Dnipro. Next slide, please. The concentration of estradiol and cortisol was determined by standard methods using standard reagents according to the instruction for the reagent set. Estradiol level was determined in Kvitko embryons, young crustacea, carcasses, samples of Procambarus virginalis, and the muscles of 1 month old crustacea, 5 month old and year-old ones. Cortisol level was fixed in gills and muscles of different age crustaceans, raw embryons, and juveniles of marvelous crayfish. The statistical analysis was performed using the program statistic of Windows. Next slide, please. It is known that reproductive processes in crustaceans are controlled by the endocrine system. These hormones play a significant role in normal growth and development. Water pollution can impair reproduction, development, immune response, and other physiological processes which can ultimately affect the survival of aquatic organisms. Next slide, please. Epigenetic programming established during development can be altered by exposure to EDCs during sensitive periods such as the preovulatory and ovulatory phases and cause immediate adverse outcomes. In addition, changes in methylation profiles and acetylation landscapes that occurs during these periods may predispose to the development of pathologies that occur later in childhood or adulthood, and more dramatically may ever be transmitted from the generation to generation. Next slide. According to the results of our research, the average level of estradiol in cave samples of Procambarus virginalis is shown on this slide. These results highlight the ubiquitous bioaccumulation of estrogens in aquatic invertebrates depending on the duration of exposure. Next slide, please. The distribution of estradiol in the muscles of Procambarus virginalis can directly and effectively reflect the effects of EDC. Xenoestrogens can act on several mechanisms simultaneously. Estrogenic xenobiotics also disrupt the physiology of aquatic organisms. Biochemically processes as triggers of oxidative stress can affect their reproductive development, cause transcriptional effects, and there are also quite powerful initiators of signal cascades from membranes. Transfer of estradiol from the digestive system of Procambarus virginalis to muscles may pose a potential risk to aquaculture, wildlife, and human health associated with the consumption of aquatic meat. Finally, the contribute to the general accumulation of estrogens in the environment. Next slide, please. Crayfish gills play an important role in the transport of respiratory gases, extraction of nitrogen compounds, and osmoregulation. The gills are the first barrier to waterborne pollutions. Gills' tissue condition is generally considered a good indicator of water quality and suitable for environmental impact assessment. Its changes can lead to a decrease in oxygen consumption and a weakening of the function of osmoregulation. A marked decrease in oxygen consumption can lead to internal hypoxia, which affects metabolism and movement. Next slide, please. Based on the results of study, it was established that the level of cortisol in the gills of Procambarus Virginalis increased depending on age. It's shown to the figure 1a and 1b. In this figure, we can see the content of cortisol in tissue, gills, muscles, roe, embryos, and juvenile of marble crayfish. Significant difference between the cortisol content in raw tissue of embryos and juvenile of the investigated crustaceans wasn't found. It can be assumed the hormone level changes in this sample are related to the direct influence of the surrounding water environment. Next slide, please. The obtained results emphasize the widespread bioaccumulation of EDCs, namely compounds with estrogenic activity in aquatic invertebrates, depending on the duration of exposure, which may indicate the role of the aquatic environment in the bioaccumulation of xenoestrogens. The study demonstrated the need to monitor exposure to EDC contaminants due to the effects on water quality, degradation, and serious toxicological effects on aquatic biota and human health to inhibit such potential. Estrogens are clearly necessary for normal fauna and human physiology, but can have serious negative consequences if allowed to accumulate in the environment. Thus, it is possible to note two important aspects related to the issue of contamination of aquatic biota with compounds that cause the destruction of the endocrine system. The first aspect concerns the impact of EDCs on the reproduction of wildlife populations. including aquatic organisms, which may have a negative impact on species conservation. The second aspect is that since the effects of EDCs are realized on humans in different ways, primarily through trophic link food chains. Further research is needed for public health policy planning. Thank you for your attention. Please send your questions and proposal for cooperation to the following email address.